Disclaimer. This presentation contains copyrighted material collected from the web resources, journals, and pathophysiology textbooks. The presentation is intended for teaching and no commercialization or marketing involved. The learners are recommended to purchase and refer the textbooks for a better learning experience.
Hi everyone, welcome to today's class on inflammation, immunity, and chemical mediators involved in the inflammation. Right. Basically, the survival of all organisms requires that they eliminate any foreign invasions such as antigens or any dust particles or any foreign bodies like bacteria, virus, or any kind of microorganisms, any infectious pathogens and damaged tissues. All these things need to be removed out of your body in a safe manner, right? So, in order to protect the body and remove such kind of things from our body, inflammation is going to take up a protective response uh, and eliminate such kind of material such as antigens from our body through certain processes, right? uh, Inflammation accomplishes its protective mission by diluting or destroying or also neutralizing the harmful agents. Acute inflammation is characterized by fluid and plasma protein exudation, and it is predominantly neutrophilic or due to leukocytic accumulation, right? So chronic inflammation is different from acute inflammation. So it is identified or it is distinguished from acute inflammation by the influx of lymphocytes, which includes macrophages and several other cells such as giant cells, etc. Right. So anyway, we'll discuss all these things one by one by specifically differentiating all the factors involved in the inflammation as well as immunity. Right. So basically, whenever we say inflammation, we think about these things: heat, redness, swelling, pain, and loss of functions. These are the common characteristics of any inflammation. So, in this presentation, you are going to understand why all these signs of inflammation are produced and what is the reason behind production of all these signs of inflammation, and we'll also discuss how inflammation is going to affect various pathological processes in the body, right? Basically, WBC, white blood cells, right? So can be divided into granulocytes and agranulocytes. Out of these white blood cells, again, there are multiple types. Again, granulocytes. Within the granulocytes, there are basophils, eosinophils, and three different further distinguished neutrophils. Right, and the commonmost neutrophils present in our body are segment neutrophils, and these are highly abundant within our blood. And apart from that, there are two agranulocytes, lymphocytes, as well as monocytes. And most of the phagocytic process in our body involves both neutrophils as well as monocytes. Right. Apart from that, immune reactions in our body are majorly generated by lymphocytes. Right. So let us now start discussing about the stimuli for acute inflammation. So basically, acute inflammation is started by any kind of infections such as bacteria, viral, fungi, or parasitic infections leads to acute inflammation. And if any trauma occurs, such as any splinter getting into our body, or anything getting penetrated into our body, or a small wound, or any kind of physical injury, or any kind of chemical injury, or in case of thermal injury also, such as burns or tick bites or irradiation. So all in all these cases, there will be stimulating the acute inflammation. And tissue necrosis would also lead to acute inflammation, such as ischemia. So, just for example, myocardial infarction leads to acute inflammatory reaction, and any kind of foreign body like splinters, dirt, or sutures, they also leads to acute inflammation. And immune reactions such as type 1 hypersensitivity reactions against any kind of environmental substances and against self tissues. They also induce acute inflammatory reactions. Right. And let us now discuss the components of inflammation. So, what are various components of inflammation and why these components of inflammation are going to produce these inflammatory reactions, and how these inflammatory reactions are going to ultimately affect various physiological processes. Right? So, the commonmost are the first and foremost uh, intermediates of inflammation are macrophages and mast cells. Right. So these are the first and foremost recognizers of any pathogen entering into our body. So once these macrophages or wandering macrophages recognizes any pathogen, so these macrophages are going to release certain inflammatory mediators into the extracellular matrix, right? So these extracellular matrix, these mediators present in the extracellular matrix are going to communicate to other cells which are present inside the blood vessels. So these macrophages releases cytokines, so which are mediators, and they have very important role in immune response. So that is the function of macrophages in immune response. And then mast cells, in response to what are the mediators released by the macrophages, now these mast cells further release histamine and some other mediators also. So these mediators would still aggravate the acute inflammatory response at the site of release, right? And then other cells include PMNs, polymorphonuclear leukocytes, nothing but the neutrophils. Neutrophils are involved in the elimination of microbes as well as dead tissues. They are particularly known for their phagocytic action. And then lymphocytes are involved in production of immune response, and monocytes are the one which turns into macrophages, whereas PMN cells, neutrophils, will turn into microphages. These are macro and these are micro. These are smaller, but still they are phasic, means they can engulf a pathogen. Whereas macrophages, these are bigger and then they can also engulf a pathogen. And then platelets. Platelets are involved in the production of thromboxanes. They can also clog the area where there is a puncture in the blood vessel. So platelet plug formation stops the leakage of blood from the area which is infected with a wound. Right. Apart from that, endothelial cells of the blood vessels are also involved in the release of certain mediators, right? So these endothelial cells releases nitric oxide and certain other cytokines as well. And there are plasma proteins present in the blood would also have a specific role on inflammation. There are specific complements present in the plasma proteins which mediates the inflammation, and plasma protein ultimately leads to elimination of microbes, means through the flow of blood. And plasma proteins also constitute certain mediators of inflammation such as clotting factors as well as kininogens. And apart from that, some other mediators of inflammation also include, are some other cells that participate in the inflammation also include fibroblasts. These fibroblasts are involved in the production of fibrous tissue such as collagen, right? So these fibrous tissue ultimately leads to covering of extracellular matrix which was abraded or which was damaged due to an injury. Right. Acute inflammation is a rapid response to injury or microbes and other foreign substances that is designed to deliver leukocytes and plasma proteins to sites of injury. You can observe that after an acute inflammation, there will be migration of neutrophils and there will be increased blood flow to the area which is infected or inflicted with a wound due to acute inflammation. Right? The capillary blood has been dilated comparatively to a normal tissue, right? And apart from that, it also leads to leakage of plasma proteins and that finally leads to edema, right? All these responses of acute inflammation are either due to repair or to inhibit the pathogenic migration towards the blood, or it inhibits the distribution of these pathogens into the various parts of blood, various parts of body, right? So it is characterized by transient vasoconstriction, which lasts for only few seconds, and then arteriolar vasodilation, which occurs for a very longer period of time. So during this vasodilatory period, so this exudation is going to occur. Then there would be redness and erythema at the site of inflammation, and it also produces warmth due to release of certain prostaglandins. And then another response of acute inflammation is stasis. Stasis means slowing of blood flow due to erythrocyte concentration. So at the site of acute inflammation, because of vasodilation, right now the pressure was reduced, and because of that, blood flow will be slowed down, and moreover, there will be accumulation of more number of erythrocytes at the site of acute inflammation, which ultimately leads to slowing of blood flow. This slowing of blood flow is very essential for the WBC to migrate out of the blood vessels. So I hope you remember white blood cells are the component of blood. They actually has to be present inside the blood. So you can observe here in a normal figure where there are very few number of macrophages and neutrophils present outside. These are called as resident macrophages. But in case of an inflammation, as you can observe, there are many number of macrophages and neutrophils have been migrated outside the blood vessels. The reason behind that is, so during the stasis of blood flow, these WBC would be able to come out of blood and reach the site of inflammation. Right? And what happens to vascular permeability during the inflammation? Let's see that. So during the normal condition, there will be more hydrostatic pressure inside the blood vessels and less colloidal osmotic pressure, right? So because of that, this hydrostatic pressure exerted on the walls of blood vessels would lead to blood blood flow in a proper manner, right? When an inflammation occurs, this hydrostatic pressure is going to increase very high, right? And the reason behind increasing for hydrostatic blood pressure is due to uh, vasodilation. At the same time, there are more number of cells over there, right? So what are the things present over here in the form of dots are proteins, not cells. Don't try to confuse, right? So here, imagine that there are more number of cells here. So there are more number of cells present over here, which makes increased hydrostatic pressure, and the fluid would leak out. And there are less number of proteins here, right? So because of less number of proteins, colloidal osmotic pressure is going to decrease. So this ultimately, this increase in hydrostatic pressure, which is commonly seen in venous outflow obstruction. So now an example for that is congestive heart failure, and decrease in colloidal osmotic pressure is seen in liver diseases, and increased protein synthesis are in case of kidney diseases, right? As well, right? Then there are two different types of fluid leakages commonly observed in the inflammation. First one is transudate. Transudate means only fluid leaks out of the blood vessels. As you can observe here, only fluid leaked out of the blood vessel. And another one is exudate. In this case of exudation, there are more number of proteins present at the site of injury within the blood vessels, and these exudate would also leak out the plasma proteins, right? So basically transudation and exudation. So an example for transudate is burn wound. In case of burn wound, as you can observe, the blister formed out of the burn wound would generally be made up of transudate. Whereas in case of any specific wound other than burn wounds, like any physical wounds or trauma, would be made up of exudate. Okay, and then this particular thing is an example of a swollen lymph node, also called as lymphangitis, right? Generally, this kind of lymphangitis is completely different, but I would like to just show you that we have two adenoids at the throat, right? So generally, these adenoids would get inflamed due to any kind of acute invasion of pathogenic microorganisms. So when the adenoids are swollen, you would also perceive that you perceive the difficulty in engulfing the food or engulfing any kind of hard fluids or anything, right? So the reason behind lymphangitis or lymphadenitis, right? So is nothing but they are giving you uh, a message, or the lymphatic nodes are inflamed because of migration of a specific pathogen into the lymph nodes, right? So in such cases, these lymph nodes, as you can observe, they will be enlarged because of hyperplasia, as well as uh, uh, mm, increase in migration of lymphocytes towards the area which was affected by migration of pathogenic microorganisms. Right. And let us now see various cellular level events at the inflammatory region. There are multiple things going to happen at the inflammatory region. Let us discuss all those cellular level events, right? There are, in fact, two different types of events happens in the inflammation. One is vascular event. So whatever we discussed is this vascular event. This whole thing is vascular event. Exudation. Exudation is nothing but vascular event. And another one is cellular event. So in this cellular event, migration of leukocytes from the blood vessels into the tissues is going to happen. So this whole phenomenon is called cellular event. So let us see how the cellular events occurs. Basically, leukocytes are the offending cells which ingest and kill the bacteria and other microbes. They're supposed to also eliminate the necrotic tissue or dead tissue and any other foreign substances. But leukocytes cannot do that by flowing along with the blood inside the blood vessels. They has to come out. They has to come out of the blood vessels, and they have to perform all these actions of engulfing the bacteria, killing the bacteria, and eliminating the necrotic tissue only by acting at the extracellular matrix, right, are the interstitial tissue, right. Now, for that particular thing, basically, what are the cells already present in the interstitial tissue are going to release certain cytokines. These cytokines, such as tumor necrosis factor, interleukins, these cytokines will be released by residing macrophages, right? And once you can observe that these residing macrophages are also already infected by microbes. They already phagocytized some microbes. If the residing macrophages are able to handle the number of microbes present at the site of injury, then they do not release cytokines. But if the load is very high, they release some cytokines to recruit more number of leukocytes to the site of injury, so that they will also participate in engulfing such kind of bacteria. Now, let us discuss the whole process involved in this recruitment of macrophages. As you can observe, there are multiple proteins present at the sites of these leukocytes. At the same time, there are multiple proteins present on the endothelial cells. Let us now discuss the role of all such proteins, which I am going to mark in red. We are going to discuss about all such proteins now in this class. Okay, right. So let us start discussing from the first thing, silyl Lewis X modified glycoprotein. So these glycoproteins are present on the leukocytes, and you can also observe integrins. Integrins are a specific form of proteins which are again present on the cell surface of the leukocytes. These integrins as well as these silyl Lewis X modified glycoproteins are very essential components of the cell surface proteins of leukocytes, which involves in the migration of these leukocytes. Basically, there are two more other important proteins present on the endothelial bed of the blood vessels. These are selectins. There are two different types of selectins present on the endothelial bed. One is P-selectin, which is commonly found on the intracellular weevil pallid bodies of the endothelial cells. Actually, these selectins are not uh, surfaced onto the cell surface. Normally, you can observe that on all other cells, the just for a diagrammatic depiction, on all other cells, these selectins are not present. Only on the activated cells, selectins are present. So when the cytokines activate the endothelial cells, then these activated endothelial cells would make the selectins to come onto the cell surface. Selectins are the interacting proteins. Selectins are particularly going to interact with silyl X modified glycoproteins on the cell surface of leukocytes. When selectins come out of the endothelial cells, these modified glycoproteins are going to interact with the selectins, as you can observe, and that leads to rolling. You can observe that the cell is rolling like this, and this kind of rolling effect ultimately makes the cell, makes this WBC to stick onto the wall of blood vessel, to stick onto the wall of blood vessel. Why this kind of rolling is required? So the reason behind this rolling is, this is just to give you an imagination of how of white blood cells compared to the population of red blood cells. You can see that there are so many number of red blood cells and very few number of white blood cells present in this small image of a which is taken under a microscope, right. You can clearly distinguish that uh, out of many number of red blood cells, there are very less number of WBC. Now, just imagine uh, a crew of soldiers marching, a crew of red blood cells are marching in the blood vessels, and along with that crew, there are few WBC which are unorganized, and they are also marching towards them. Right. In that case, you can just clearly understand the domination of red blood cells on the flow of blood. So, because of that such kind of domination of red blood cells, basically WBC are always going to roam on the edges of blood vessels, whereas red blood cells would move on the center of blood vessels. Right. So, this also has a specific benefit because on the edges of blood vessels, when the WBC are moving, when they can identify such kind of selectins, then they particularly activate and they can start rolling, and finally they are going to integrate, right? And then there is another specific protein which is also surfaced on the cell surfaces of endothelial cells. Uh, that particular protein is ICAM, integrin ligand. Right. This integrin ligand is a specific protein binds to the activated integrins on the cell surface of leukocytes. You can observe this is the form of inactive state, and once activated, you can observe this activated form would be like this. So once activated, it can now bind onto the integrins. The difference between selectins and integrin is selectins can maintain some kind of contact uh, between the endothelial cells and the leukocytes, and they ultimately leads to rolling effect. Selectins leads to rolling effect, whereas integrins lead to integration, means they lead to binding effect. So as you can observe, when there are more number of integrins and selectins ultimately bound onto the cell surface. So this ultimately led to stoppage of this WBC. This WBC is now struck over here. It cannot flow anymore. It cannot flow along with the blood anymore like this. It has to struck over there. So once it is struck over there, then there are some other platelet endothelial cell adhesion molecules, PECMs. Platelet endothelial cell adhesion molecules would erupt out of the leukocytes. These platelet endothelial cell adhesion molecules will make the gap between the endothelial cells, and ultimately this lead to migration of a uh, neutrophil or a macrophage. And then once it comes out of the blood vessels, now the next thing is chemotaxis. Chemotaxis means movement, right? Now let's say the pathogen is present here, and now in order to engulf these pathogens present over here, this monocyte, this neutrophil has to move from here to here, right? So such kind of movement is only possible only when the cell has some certain pseudopods, right? So there are certain pseudopod kind of structures present on these lymphocytes and monocytes as well as neutrophils, right? So these pseudopod kind of structures would make the cells to migrate towards the area which was inflicted with a wound or inflicted by pathogens and all, and they are going to start producing their action, right? Apart from that, you can also observe that after the migration of these cells. So the process of migration is called chemotaxis. Chemotaxis. So after the cells are migrated, there would be two different types of events going to take place. So predominantly for the first 6 to 24 hours, there will be major migration of neutrophils. As you can observe, these are granulocytes, isn't it? Right. So there are more number of neutrophils at the first 6 to 24 hours. Later on, after 24 hours, if the inflammation is not subsided, that means the pathogen is stronger. In that case, after 24 hours, around 24 to 48 hours, after 24 hours generally, there will be migration of monocytes are observed. As you can observe, there are many number of monocytes here, right? And there are also certain neutrophils, right? There are certain neutrophils would be present, but from then onwards, you can observe that monocyte migration would be specifically increased, right? And yeah, the same thing has been depicted here also. uh, on the first day, you can observe that edema is going to occur excessively, and on the first day itself, neutrophils also migrate. And from the second day or third day onwards, monocytes and macrophages would start migrating towards the area of inflammation, and their activity is going to increase accordingly. And then this is a process of phagocytosis. So once the microbes are identified by the macrophages, they are going to engulf the pathogens, and once the pathogens are engulfed, these pathogens would be forming a phagosome, and then later they are going to combine with a lysosome, which finally forms a phagolysosome. So within the phagolysosome, there are lysosomal enzymes, um, which ultimately leads to killing of pathogen through the generation of reactive oxygen species, and ultimately this and also there are certain enzymes like myeloperoxidase which leads to production of nitrous oxide radicals. This ultimately leads to killing of the pathogen within the phagolysosome. Once the uh, pathogen is completely dead within the phagolysosome, further the remnants of this phagolysosome will be released again onto the extracellular matrix. So this is a brief introduction of inflammation. So let us now start discussing about the chemical mediators of inflammation. So till now we discussed about the whole process of inflammation. Now let us start discussing about the mediators of inflammation and what is their role and how these mediators of inflammation are going to change the way the inflammation is going to proceed. All these things will be the part of uh, this particular part of lecture. Right. So chemical mediators of inflammation such as histamine. Histamine is released from mast cells and basophils as well as platelets. The effect of histamine is vasodilation, increased vascular permeability, and endothelial cell activation. As you can observe in this process, so let's say this is a mast cell. So this mast cell releases histamine, which activates the endothelial cells, which also dilates the blood vessels. So basically, it is very important mediator involved in the inflammation. Then prostaglandins. Prostaglandins are released from mast cells and leukocytes, and they are involved in the vasodilation, and they cause pain, and they also produce fever. And leukotrienes. Leukotrienes are produced from mast cells and leukocytes again. Leukocytes particularly release leukotrienes in case of acute inflammatory reactions such as asthma, and they cause increased vascular permeability. They lead to chemotaxis, means movement of these neutrophils and macrophages from the blood vessels outside the blood vessels, and leukocyte adhesion and activation. So as you can observe, leukotrienes are responsible for many actions to mediate the inflammation. And cytokines such as tumor necrosis factor, interleukin 1 and interleukin 6, these are very, very important mediators of inflammation which are particularly released by macrophages. As you can observe, till now macrophages have never entered. So macrophages releases such mediators. So these are very important messengers which activate the endothelial cells, and they also lead to expression of L-selectin and P-selectin, which we discussed earlier, right? And these mediators would also cause systemic effects such as fever and metabolic abnormalities, and shock would be produced due to hypotension. Uh, and these are the common effects of cytokines. And then chemokines. Chemokines are produced by leukocytes and activated macrophages, and they cause chemotaxis. So in this figure, which we discussed earlier, these are the chemokines. Chemokines are the messengers which will activate the macrophages or microphages, and they give a message to the microphages in such a manner that they need to migrate outside, right? So they just give a message. They act as messengers, and they ultimately leads to chemotaxis. So chemokines are involved in the chemotaxis and leukocyte activation. And then platelet activating factor. Platelet activating factor is released from mast cells, so which leads to activation of platelets, the name itself implies, and it is also released by leukocytes. And further, it also causes vasodilation, it increases vascular permeability, and it increases leukocyte adhesion, and it produces chemotaxis, degranulation of mast cells, oxidative burst, and most importantly, they need to be present to activate the platelets. So once the activated platelets, that also releases thromboxanes, which further causes vasodilation. Right? And then complements. Complements are a part of plasma. Complements as well as kinins. These are the part of plasma which was actually produced by liver. Right. So complements are involved in the leukocyte chemotaxis and activation. So I will explain the actual function of complements in the later half of my presentation. These complements are involved in opsonization. Opsonization means recognition of a pathogen. And complements would also lead to direct killing through membrane attack complex, and they also cause vasodilation through mast cell stimulation. So we'll further discuss about these complements in the next few slides. And then kinins. Kinins are produced by liver itself into the plasma, and these kinins are also involved in increasing the vascular permeability, and they cause smooth muscle contraction, vasodilation, and they are mediators of pain as well. So these are certain principal mediators of inflammation. And then this figure is going to give you an idea about how certain mediators leads to various types of activations. You can clearly observe there are two different kinds of activations. One is classical activation of a macrophage and alternative activation of a microphage. So both are macrophages only. There is no difference. These two are macrophages only. But their activation was differed, right? Activation was differed. So an activated macrophage, if it releases interferons, so this activation leads to activation of inflammation, such as, so when an activated um, macrophage releases interferons, it leads to release of ROS, nitric oxide, lysosomal enzymes, which would ultimately leads to phagocytosis of many bacteria and fungi. At the same time, other macrophages also releases interleukin 1, interleukin 12, interleukin 23, and some other chemokines that activates the process of inflammation. Whereas the same macrophages, if in turn, if they release some other, such as interleukin 13 or interleukin 4, interleukin 13 and interleukin 4 are the inhibitors of inflammation. They are going to inhibit inflammation and they activate repair. they activate repair mechanisms. As you can observe, when interleukin 13 or 4 are released by the macrophages, other macrophages in response to it, they release TGF beta. It is a growth factor, means tissue growth factor, which is involved in the tissue repair and fibrosis, and it further releases TGF beta as well as interleukin 10, which also have anti-inflammatory effects. So these two, interferon gamma and interleukin 13-4 released by the macrophages, in turn either activate or deactivate the inflammatory mechanism, either activate the inflammatory mechanism or activate the anti-inflammatory mechanism. Okay, which means either activate inflammation or deactivate inflammation. Okay, so then let us now discuss about various mediators of inflammation in a large context. Let us see how these mediators of inflammation are involved in various physiological processes. Right. So whatever we discussed already, histamine is released from the mast cells. Histamine is stored in the form of granules, and as well as serotonin also. These two are stored in the form of granules, and mast cells as well as platelets are going to release histamine as well as serotonin. And once the mast cells, basophils, platelets are activated, then they lead to release of histamine or serotonin. Then the major action of either histamine or serotonin is to initiate the acute inflammatory reaction, and particularly histamine is going to produce the vasodilation, which is an important vascular event which happens at the beginning of inflammatory response. And apart from that, I hope all of you know about these things: prostaglandins, leukotrienes, platelet activating factor, right? So all these things are released by inflammatory cells, uh, which was, I hope, widely studied by everyone, right? So prostaglandins, leukotrienes, lipoxins are very important mediators of cell-derived inflammation, right? So basically, these products are produced from arachidonic acid. From the cell membrane, phospholipases are going to liberate the arachidonic acids, so which is an eicosanoid. So this arachidonic acid would be further processed by two different enzymes such as cyclooxygenase or lipoxygenase to produce prostaglandins or leukotrienes. So prostaglandins such as prostacyclin, PGI2, thromboxane A2, PGD2E2, all these prostaglandins are produced by cyclooxygenase pathway. Now, out of these prostaglandins, some of the prostaglandins are the initiators of inflammation, and some of them are initiators of repair or anti-inflammatory. So, just for example, prostaglandin D2 and E2, they cause vasodilation and they increase vascular permeability. And thromboxane A2 causes vasoconstriction and promotes platelet aggregation. So, as you can observe, the effects of PGD2, E2, and thromboxane A2 are completely opposite, right? Further, PGD2 and E2, they involve in inflammation, whereas prostacyclin PGI2 is involved in repair. They cause vasodilation, but still they inhibit the platelet aggregation. So once the platelet aggregation is inhibited, inflammation would be slowly subsided. And then some other mediators include leukotrienes, right? So leukotrienes are particularly involved in the immune reactions. They are going to cause leukotrienes are specifically involved in respiratory inflammation or bronchospasm. Means in response to the leukotrienes, bronchial smooth muscle is going to constrict, and because of constriction, it causes bronchospasm, and person wouldn't be able to breathe. So especially in asthmatic attacks, leukotrienes are seen to be activated highly. So anti-leukotriene drugs are leukotriene antagonist drugs such as montelukast are administered as antagonist of here. And lipoxins are involved in the anti-inflammatory effects. So they are going to inhibit the inflammation. So all these are various inflammatory or anti-inflammatory mediators further released by the uh, cell-derived proteins only. Apart from that, as you can observe, all leukocytes would also produce ROS, reactive oxygen species, to kill the bacteria and nitric oxide as well. And cytokines are produced by macrophages, lymphocytes, and mast cells, right? So all these cytokines are involved in recruitment of um, further leukocytes or macrophages, and neuropeptides, which are released by nerve fibers and leukocytes, are involved in the pain perception. So, such as substance P induces pain, right? So, these are some other factors. Apart from that, um, some other components include uh, complements. Right? So we are just going to see what are complements. I'll explain the exact action of complements in next few slides as well. Complements produced from the liver, which is released into the plasma, and there are some other kinins and coagulating factors also released by the liver into plasma. They also have specific inflammatory properties, and their actions will be further discussed in the next part of presentation. And then let us discuss about cytokines. Cytokines are very important inflammatory mediators. There are different types of cytokines involved in early immune response, and some of the cytokines are produced in the later responses, means in producing a repair as well. So, uh, right, let me explain about some of the cytokines which are commonly observed or commonly renowned. Tumor necrosis factor, interleukin 1, interleukin 6. These are some of the common cytokines which are involved in inflammation. Let us see their actions. So, TNF as well as interleukin 1 are involved in the activation of selectins on the endothelial surfaces, and TNF also increases the permeability by producing endothelial contraction. And endothelial cells, once activated by TNF and IL interleukin, they further release interleukins and chemokines into the interstitial tissue. And TNF and IL-1 leads to activation of leukocytes as well as macrophages, right? So leukocytes such as neutrophils as well as macrophages would be involved in further release of some more chemokines, some more loads of chemokines and interleukins, and they have systemic protective effects. So these cytokines are going to produce fever. They are going to affect the hypothalamus in the brain, and they lead to fever. And certain acute phase proteins are produced. Acute phase proteins are nothing but, as we discussed earlier, complements as well as kinins are produced by, or the production of kinins and complements will be activated by interleukin 1 and interleukin 6. And further, they also activate the bone marrow to produce more and more number of leukocytes. Reason is because during the process of inflammation, leukocytes are being recruited, neutrophils are being recruited to the site of injury, and these neutrophils are ultimately getting killed due to the pathogens. So because the killed microphages or macrophages need to be replaced with new sets of leukocytes, so leukocyte production will be initiated by stimulation of bone marrow, but they also have some pathologic effects. Tumor necrosis factor is going to decrease the cardiac output, and they also leads to thrombus formation. So thrombus formation could clog the blood vessels or reduce the blood flow, and that may also cause certain heart attacks, as well as tumor necrosis factor and interleukin 1 also causes insulin resistance, and they make the muscles weaker, right? So skeletal system becomes weaker when a person is affected with inflammation, right? So all these effects are produced by chemokines, sorry, cytokines, right? So the exact effects of cytokines at various sites. So they are going to increase leukocyte adherence. They are going to increase the prostacyclin synthesis, which is a vasodilator, and they cause procoagulant activity. And they also have anticoagulant properties. And they still leads to release of further cytokines such as interleukin 1, 8, interleukin 6, and platelet-derived growth factor. And they produce certain systemic effects, so they cause fever, they increase sleep, they reduce appetite. So basically, we need to discuss further about this reducing appetite. So when a person is affected with an illness, right, our body tells us not to take especially our tongue feels tastelessness, right? So and our body feels weaker, and it tells us to sleep, right? But anyway, if we try to disobey our own body's response and try to do opposite to what our body says to us, then it makes us further weaker, and makes us to suffer from the pathogenic effects. So, in order to uh, basically, all these cytokines are producing these kind of responses in order to make the body recover faster. So, while you are sleeping, your body is going to recover quickly, and it makes the appetite to lose because it doesn't want to feed the pathogen. So when you are still taking the food, you are not only feeding your own body, you are feeding the pathogen itself, right? So these are some of the signs given by our own body to protect us from the pathogenic effects. Then acute phase proteins are increased, nothing but complements and kinins from the liver are increased, and hemodynamic effects such as shock are produced, means um, there will be acute loss of fluids, and there will be um, also cardiac strokes which would commonly occur, and shock means increased blood pressure and increased heart rate, right? And neutrophilia occurs due to tumor necrosis factor and interleukin 1, because many number of neutrophils are being recruited to the sites of inflammation. That's why neutrophilia, number of neutrocytes in the blood decreases. And other fibroblast effects include increased proliferation of fibroblasts. So let's say if there is an injury on a part of body like this, so this site which is injured has to be filled with some space, right? So the space filling component is fibroblast. Fibroblasts are going to release collagen, and they fill the space which is inflicted by the wound. So cytokines are going to activate the fibroblast to fill up the lacuna, produced by the physical wound. So later, this lacuna would be replaced by the actual parenchymal cells which has to be present over there. So basically, it should be skin cells, so such as epidermal cells, right? Then they also leads to increased collagenase, protease, prostaglandin E synthesis. All these things will be effects of fibroblasts. And then leukocytic effects includes increased cytokine secretion such as interleukin 1 and interleukin 6. So these are certain effects of cytokines. And then, right, something that I've been telling from very long, complements. So what is the function of complements? Complements are involved in a very important process in the inflammation called opsonization. Opsonization. Opsonization means flagging. You can also remember this as flagging, right? Flagging, right? So what is flagging? Right? Basically, any pathogen once enters into the body has to be detected by our immune system, and our immune system has to recruit many other immune cells to kill that pathogen, right? But all our immune cells would not be able to recognize the pathogen, right? So the process of making a pathogen to get recognized, the process of making a pathogen to get recognized is nothing but opsonization, right? So in a classical pathway, once a pathogen enters into our body, antibodies which are already present in our body against that particular pathogen are going to attach to the pathogen and makes the pathogen to be identified. This is one kind of complement activation. And then lectin pathway. So in this pathway, there are certain mannose binding sites present on the cell surface of the pathogen, such as any bacteria, and onto that mannose will bind and makes this bacteria to be visible to many other inflammatory cells, right? So in this manner, ultimately, once the cell is identified, then certain complementary proteins such as C3b and C3a will bind onto the cell surface, or they will be deposited on the microbe. So once they bind over there, a fragment of complement C3a is released out of this attached complement, and this C3a would then go and complain to the neutrophils. So this leads to recruitment of more number of neutrophils, and this ultimately comes and detects these pathogens and engulfs the pathogens and kill the pathogens. And then it could also lead to complaining. I mean, these complements act as uh, uh, recognition sites by some of the phagocytes, so such as macrophages identify any cell or any body which is attached by the complement as a foreign body, and then they will engulf that and they can kill it. Apart from that, complements they themselves leads to killing. Some of the complements leads to formation of membrane attack complex (MAC). This membrane attack complex leads to formation of certain pores which makes the bacteria to leak its cellular contents. So because of that, microbes are going to get lysed. So these through these mechanisms, ultimately complements are going to opsonize as well as protect our body from the pathogens. And then coagulation factors as well as kinins produced by the liver into the plasma. Basically, Hagman factor, factor number 12, which is produced by the liver into the plasma, gets activated by high molecular weight kininogen, also known as kallikrein. Kallikrein, which is produced in the process of cascade coagulation cascade, is going to stimulate further formation of many other complements. You can just go through this process of coagulating cascade later on. I hope all of you would have come across this during your bachelor's degree. So the cascade of coagulation is going to ultimately stimulate the formation of kallikreins, or kallikreins further stimulate the formation of plasmin, and that ultimately leads to activation of complements, which we discussed here. Complement C3A. Complement C3A would be activated by the coagulation cascade as well as kinase systems. So this ultimately protects or makes the pathogens to be recognized by the uh, microbes. Sorry, by the complements. And in a summary, let us discuss the roles of all these mediators. So, which we just discussed, we'll discuss the roles of all these mediators. There are three different chemical mediators involved in producing vasodilation: prostaglandins, nitric oxide, histamine. So, prostaglandin E2 and D2 produces vasodilation. Prostaglandin I2 also produces vasodilation. But prostaglandin E2 and D2 are inflammatory, whereas prostaglandin I2, also called as prostacyclin, this is anti-inflammatory. Okay, and nitric oxide is going to produce vasodilation, which is particularly released by endothelial cells. It is also called as EDRF, endothelium derived relaxing factor, EDRF. And apart from that, histamine also causes vasodilation. And then increased vascular permeability is stimulated by histamine and serotonin, complements, and bradykinin, leukotriene C4, D4, and E4, platelet activating factor, substance B. All of them are involved in increasing vascular permeability. And then interleukins and tumor necrosis factor leads to leukocyte recruitment and activation. Chemokines, complements, LTB4, all these things leads to leukocyte recruitment. And prostaglandins and interleukins causes fever, and prostaglandins, bradykinin, and neuropeptides could also induce pain, as well as lysosomal enzymes of leukocytes, reactive oxygen species, and nitric oxide. All these are going to injure tissue. At the same time, they also lead to killing of pathogen. So within the phagolysosome, the pathogen will be killed by all these things, including lysosomal enzymes, reactive oxygen species, and nitric oxide. So all these things kills the pathogen inside the phagolysosome. So these are the roles of all mediators involved in inflammation. So with this, we'll break the class, and the next set includes immunity. Thank you.